just as usually, advertisement to the next week. It will be about the way to kitchen. In fact, more about cockroaches and their way to other kitchens, about synanthropy of cockroaches worldwide. It's all given by Zuzka Kotyková-Varadinova. So you are welcome in a week as well. And today we have a little bit different topic. And it's my big pleasure to welcome here Antonia Weberling. Antonia is embryologist. She started to study human and mice and such like boring species, and then she switched to something more interesting, which are reptiles. So she studied PhD in Cambridge, and then she switched to Oxford, and yeah, she will tell you more. So welcome.
Do you mind closing the window? Yeah. Hello everyone, thank you very much for having me. Lukas said, my name is Antonia, and this one here. I'll quickly introduce myself, and then I will go into our talk. So I'm a biochemist by training. I did my bachelor and master's in Berlin before I moved to the UK to focus during my PhD on mouse and human early embryo development, so pre-gustulation. And then I moved to Oxford, where I was awarded a 5-year fellowship to establish my independent research group. The work that I'm doing is largely collaborative, and I wouldn't be able to do anything without my amazing collaborators that have all of the animals that I want to have access to. So the stories I'm presenting you today are in collaboration with Professor Paul Trainer and Natasha Schüler from Star Wars Institute. Natasha is just about to start her own group at Rowan University, and Professor Tom Zenger at Loyola University in Chicago, as well as my postdoc supervisor Florian Holfelder in Cambridge, who were able to already start the project before I started my fellowship. So, why do we want to study early embryos in the first place? Some of you might know this, this graph. It's called the pregnancy loss iceberg, which shows us that 60% of human pregnancies fail in the first 2 weeks of development. 30 of them don't make it past implantation, and another 30% don't make it for the second week, which ends in the time of gustulation. Generally, 1 in 7 couples in the UK is infertile. The tendency is rising. So what happens in these first 2 weeks that is so essential that these embryos spontaneously abort themselves rather than continuing development? And I will now guide you through these weeks and show them in mouse and in human embryos, and then continue with that. So embryogenesis, you have here mouse and human embryos next to each other, but I will focus on the human side of things. Embryogenesis commences with the fusion of the male and female gametes into the totipotent zygote, which then undergoes a series of cleavage divisions, where you have loosely adherent blastomeres until the 8 to 16 cell stage. At this time period, the embryos start compaction, so they start building tight junctions and adhesion and cell adhesions with each other until they form one tightly linked ball of cells, the morula, which is still inside the zona pellucida that you see here as a shadow, which prevents ectopic pregnancies. At this time period, all of these blastomeres have the same potential. At this time period, now these blastomeres can define an inner and an outer surface, because the outer surface is facing the zona pellucida, the inner surface is going to face another blastomere. And this enables, during the next cleavage divisions from the 16 to the 32 cell stage, that the first lineage segregation happens, which parts the outer cells that you see here in blue to become the trovectoderm, the future placenta, and the inner cells that you see here in red to become the inner cell mass. Once this segregation is completed, these cells start to pump fluid and inflate and form a lumen and give rise to the so-called blastocyst, which eventually then hatches out of the zona and is ready to implant. The formation of the blastocyst enables the second lineage segregation that you see here happening, where the red cells, which are now becoming the epiblast, which is the actual body of the embryo, part from the yellow cells, which is the hypoblast, which are going to become the yolk sac. Which, again, upon hatching, these embryos are then ready to implant. And now I quickly have to get away from the presentation and show you the video, the live imaging of it. Here you see that now in real life. This is a mouse embryo. You see the 2-cell division, you 4 cells, and then towards 8 cells. And now they are compacted, and in a second you will see the inflation happening here. So that is the, unfortunately didn't play here. Yes. So that thus far for pre-implantation development. Then the embryo has hatched from the zona pellucida and is ready to implant. And upon implantation, the embryo has to undergo a series of maturation and tissue rearrangement events. Any of these maturations or tissue rearrangements going wrong would lead to a spontaneous abortion of the embryo. So we'll focus here first on the human and then on the mouse, because you can see already from the drawings that they start to become quite different. So the human embryo implants from the polar trophectoderm, which is in darker blue, which is overlaying the inner cell mass. And at the same time, and this is roughly around 6 to 7 days post-fertilization, and at the same time, the epiblast starts to polarize. So in the beginning, these cells, I drew them without a shape, they actually are not polarized at that time. So they are naive pluripotent, and upon implantation, they start to exit naive pluripotency and become susceptible to their surroundings and define a basal and an apical surface, and thereby form a monolayered epithelium surrounding a central site, which then they secrete negatively charged vesicles towards the apical surface, which leads to an opening of the lumen via charge repulsion, which then leads to the opening of the amniotic cavity. The cells, the epiblast cells that directly underlie the trophectoderm here, are going to become the amnion then, while the lower cells are going to become the actual body. In the mouse embryo, this is now quite different, because the mouse embryo doesn't implant from the polar trophectoderm side, it implants from the opposite end. It implants from the mural trophectoderm, which then leads to a very different overall shape of the embryo. You can see here that the embryo doesn't become long and flat, but instead becomes a cup shape, and that the trophectoderm tissue here does not stretch out in every direction, but instead folds over to form this part of the X-cylinder. And then we have two very different structures. At the same time, the lumen opening of the epiblast itself and the exit of naive pluripotency are very similar between these two. So I'm going to show you here 3 movies of embryos that are just folding into form this mature X-cylinder. And you see here the epiblast, this fainter ring in the inside. The large part here, the large tissue here, is the hypoblast, or the visceral endoderm. And all of these cells up here are the trophectoderm, so the future placenta. And now these cells invaginate and push the epiblast down into the visceral endoderm to then form the X-cylinder. So you can see now all of these going down versus while the embryo is growing towards the dorsal side. And then eventually you have the full X-cylinder. And you have here, these are all different embryos, but you can see in each of them quite nicely how now here it's completely invaginated also here until the embryo is then complete. At this stage, So once the X window is formed, the embryo is radially symmetric, which we have here now. And I will now first explain the mouse embryo and then eventually go towards the human, because at this time period we come to a point where we don't know that much about human embryos anymore. Because upon the time of implantation, if they are transferred, or in vitro culture of human, if they are IVF embryos, they will be transferred to the mother and thereby be withdrawn from direct observation. So just after implantation, the trophectoderm here, so the future placenta, is secreting BMP4. BMP4 in turn activates Wnt3a nodal in the epiblast. And that has a gradient, so it's most highly expressed close to the border and lowest expressed at the distal tip. This gradient activates the bottommost cells of the hypoblast, so of the visceral endoderm, the future yolk sac, to start to express antagonists against Wnt3a nodal. These two antagonists are lefty and Cerberus. And thereby, these cells down here specify into the distal visceral endoderm. Then, these cells start to unilaterally migrate towards one side, towards the border of the trophectoderm and the epiblast. And here they stop, and then form the anterior visceral endoderm. They still continue to express lefty and Cerberus, which inhibits BMP4 and Wnt3 and nodal, which are posterior markers, and thereby they lay out the anterior-posterior axis of the embryo. So just overlaying the anterior visceral endoderm is where the head is going to form. At the most opposite end, so on this side here, this is where gastulation is going to happen. So this is where the cells are going to ingress to form the mesoderm and the endoderm, to then have the three germ layers that form the entire body. This is quite well studied for mice. For humans, we don't know that much yet. We were able to, during my PhD, I was involved in a few projects where we were able to figure out that we do have an anterior hypoblast signaling center. But if you just look at the tissue architecture, there are some things that cannot be exactly the same. Because here the BMP4 is secreted into this cavity. If the trophoblast, so the blue cells here, would secrete BMP4, it would never reach the amniotic cavity, because there's an amnion in between. So it might actually be that we have the same signaling sources, but different signaling centers. That's something we don't know yet. So I'm now going to show you a movie of this anterior visceral endoderm migration that you can see here. The cells slowly moving up and always following a lead cell. The same is visible now for human embryos. Here you can see a 9-day-old human embryo that we cultured in vitro, and it's stained for Cerberus in green. You can see here the epiblast, so the actual embryonic tissue, and then we only have on one side Cerberus expression, and with some quantification that indeed these cells always cluster towards one side, showing that we do have an anterior hypoblast signaling center that is similar to what we find in mouse. So following anterior-posterior patterning, the embryo is then ready to gastulate. And at gastulation, as I said before, this is happening at the far end from the anterior hypoblast, so at the opposite side. And at this side, these cells are going to break through the basement membrane, which will be formed with collagens and laminins, and become mesodermal, ingress, express the mesoderm marker procuri, and then migrate between the epiblast and the hypoblast along even up to the other side. And here we see now a cross-section along that line here roughly of a mouse embryo. And here you can see, I would like you to focus on this part here. This is where gastulation is going to happen. And you can see that every now and then there are some small cells that are flipping over until this side gets thicker and thicker. And you can see that there is a double cell layer forming of now elongated nuclei in this direction. You can see here. this part. So, yeah, this is up to gustulation. This is pretty much how far I'm interested in. And it's something I try to answer in my PhD quite a bit. How much mouse is in each of us? Because we commonly use mouse embryos to model human embryo development, because mice are also mammals. But when we compared them, we actually found that they're not that similar. So here you can see the activity of different signaling pathways, and you can see that the time point of implantation creates quite a switch point, which also explains why so many human embryos die at the time point of implantation, because so many different signaling factors need to be switched on or switched off at that moment in time. But if we inhibit them, we can see that just looking at the anterior-posterior patterning, that human embryos need BMP signaling to be continuing to be upregulated to not only specify the anterior hypoblast, but to keep it. Mouse embryos don't. Just one of the factors. Notch signaling is very important for human embryo survival. It is not important for mouse embryo survival. So even though we commonly use mouse embryos for the humans, they actually are quite different. So, so I started to ask myself, what happens if we zoom out a bit further? So up here we have the mouse and the human, but there are a lot of different shapes in mammals actually. We have rabbit embryos here that get rid of the trophectoderm and don't form a lumen at all. We have the elephant shrew, where the inner cell mass ingresses somehow from a pluriblast. We have the tenrec, where we at least know which part of the blastocyst ingresses. In marsupials, it's a bit similar to the tenrec. Part of the blastocyst is pluripotent and is able to form the epiblast and the hypoblast, but again, we don't have a lumen. And down here we have monotremes that form a single cell layer of interspaced epiblast and hypoblast cells, which then form a two-layered embryo. So what I try to answer in my research is, how are all of these different shapes achieved? If all the signaling pathways that govern the first lineage segregations, epiblast epithelialization, and anterior-posterior patterning, as well as the initiation of gustulation, if they are all exactly the same, then how do we have this plethora of differences? This brings me to the next part, which is that we know of this massive divergence in mammals, but if you open the developmental biology textbook, it will tell you at some point that all reptiles develop like chicken, and the chicken develops like this. And here you can see a fast forward through the first day of chick development. You have cleavage divisions, you have a mass of cells that thins out to form a monolayered epithelium. There you have hypoblast cells migrating beneath, and then this structure undergoes anterior-posterior patterning and then eventually gustulation. So this is how it looks like in the original staging series. We have here a mass of cells eventually the anterior-posterior patterning developing, which becomes more clear at this stage. And then at this stage here the egg is laid. And then now the next staging series is the post-oviposition, and here you can see very nicely how the primitive streak is forming. That is the site of gustulation. So, do really all reptiles develop like chicken? Just as a quick overview, the reptile-mammalian divergence happened 220 million years ago. The avian and non-avian reptile divergence happened 250 million years ago. There are over 25,000 reptile species, and there are around 6,400 mammalian species, but still, which had, as I already described, a massive amount of divergence. But still, all reptiles are supposed to develop like one specific species. How did this concept come to pass? And I'm sure you all are very familiar with the images I'm going to show you now. I think this is due to the model organisms of biology. We would have C. elegans, Drosophila, the zebrafish, xenopus, chicken, mice, and in case you want to do something with brains or very human-specific, you would have octopus and non-human primates. And generally, chicken are used as a model for the reptile clades, but in reality, these organisms were not chosen because they are representative of their respective phylum. They were chosen because they are small, they have a fast generation length, and they're easy to handle. Chicken are easier to breed than crocodiles. So unfortunately, we have more knowledge of chicken than on crocodiles. So I will now repeat two of the canonical differences between mammals and reptiles, which I will then challenge in the rest of my talk. So generally, mammals are thought to have a three-dimensional embryo development with an epiblast lumen, and we have an amnion formation that forms pre-gustulation. Reptiles instead are generally thought to have a flat, monolayered epiblast disc, and we have an amnion initiation that happens upon neurulation. I would like now to introduce you to the model clade I'm looking at, the squamates, which have a bit over 9,000 species, and we're looking at two different, the veiled chameleon that you see down here, and the brown anole for the rest of the talk. For the brown anole, this has been a collaboration with Tom Sanger at Loyola and Paul Trainer at Stowers Institute in Kansas City. With Tom, we meet usually, even though he sits in Chicago, we meet once a year in Florida to catch lizards. And then the lab work and analysis for the project I'm showing you here has been done in the lab of Paul Trainer at Stowers Institute. The second part of the project is now done, has been partly done in Chicago and partly in Germany and in Göttingen. So the genus anole has become a model clade for adaptive radiation. We have roughly 400 individual species found in the Caribbean and Central and South America. The pre-oviposition period spans from fertilization up to limb bud formation stages, and that has never been described. Tom has staged the post-oviposition development, but yeah, there was nothing known for the pre-oviposition development. What we do know is that they lay one egg per week, roughly, from May until early September. Yeah, so how go about this? You go to Florida, you set up a lab, and you start catching lizards. You have to catch the right species. This is actually a green anole. You didn't want that one, you want this one. You tried to avoid the bigger ones and appreciate the nature and the birds and everything else around you while not being eaten by crocodiles. So this is me catching a lizard. We do that with fishing rods, where we have a small, you can see this here, a small nose attached that it's like a small lasso that you place around the neck of the lizard and we pull. You see me here very proudly with my very first lizard I ever caught. To be fair, I spent most of my time in Florida in the lab, and Tom caught the lizards. So you dissect the eggs from the female abdomens and then transfer the eggs into PBS, and then dissect the embryos inside PBS, perform a rough staging, and we fixated them in PFA or glutaldehyde, and then in the next morning we transferred them into methanol or ethanol, depending on what we wanted to do with them later. And I will now show you the staging series that we wrote that has just been accepted in developmental dynamics. The embryo starts quite similar to chicken, actually. We have the cleavage divisions here. You can see here the first four. And then we have the first blastomeres forming, but still large cleavage furrows, until eventually the entire embryonic disc is full with small cells. And if you look at this on SEM, you can see here very nicely the first cleavages, but you could already see the initiations of the 4 to 8 cleavage division. You can see here and here. So this is the beginning of the furrow, and on the opposite side you see a hole, which is going to make the next cleavage furrow. And at later stages, You have very small blastomeres at the inside, larger ones at the outside, and we have a multicellular embryo layer here. This enabled us to describe 5 stages of fertilization: the initiation of cleavage, the first blastome formations, until eventually everything is full with blastomeres but of different sizes. Until then, everything is filled with tiny blastomeres, but they're not coherent yet. If you dissect them, the embryo will rip apart, so you always will have to take a plate of yolk beneath. In the next stage, then the embryo becomes a cohesive mass, which is here. So we have now a sheet of cells, which is actually quite similar to what you would expect from chicken, with the difference is that chicken overlays a lumen. The anoles overlay the yolk. There's nothing in between the embryo and the yolk. And interestingly, then I expected that we would just have, at some point, this sheet undergoing anterior-posterior patterning and a streak or something appear and a blastopore. But instead, the embryo started to hollow. So it started to bulge out. And up here we have the dorsal side, so the top view. And this is the side that faces the yolk. And you can see here it really is hollow and it becomes even stronger. Until eventually, now here is the posterior axis and here we have the site of gastrulation. So it doesn't quite look like chicken. And here we can see this now in with Darby staining, so DNA. And you can see again just the sheet, then how it becomes dome-like, how that becomes even stronger until then we have gastrulation happening. Which leads to another 5 stages. Again, we have that hollowing that has not been shown for chicken, until we then go towards neurulation. And at neurulation, it is also stemming from that hollow structure in the beginning. It's also quite different from chicken because that embryo from the anterior, as you see here, to the posterior is bent. In the chicken, that would be straight. And then that undergoes neurulation, so these 2 sides come close to each other. The embryo closes in the middle and then zips up into either direction. You can see here a bit better. Again, we have here the blastopore remaining, and then we have the closure in the middle, and then they zipper into either direction. And again here with the stages. Until then, eventually we have neurulation, the completion of neurulation and the initiation of organogenesis. You can see the eyes developing, the heart becoming more pronounced, and the inner ear. And also the pharyngeal arch is developing, and the eye going from just the butt into now showing the lens as well. And there are another 5 stages, but I will spare you these. And instead show you what we can do with them now. So we now have a staging series which will allow us to actually look at defined stages. Now we need better resolution to be able to look through the entire embryo at single cell and do and in-depth imaging. The answer to all of these requirements is my new collaborator Jan Huskin, who has built the so-called flamingo microscope. It's a novel type of light sheet that has a modular building block system that enables you to adapt the microscope for specific needs, and it also is very small, so you could take it to field work, which is very nice. And here you see one of our first embryos, and I hope the movie is going to play. Yes. And you can see here that we have a very nice 3D view and single nuclear resolution that you see here in blue. So this is a first look at anole gastrulation. Here we see Darby, so DNA, and here brachyury. You can see already the 2 mesodermal wings, but you can also see the streak extending in this direction. And that is coinciding with the breakage of the basement membrane, which you see here in magenta, which is laminin. To conclude, we have a perigastrulation development and initiation of neurulation in anoles that diverges quite a bit from chick development. We have this hollow dome morphology that I've described. And we So where you have the embryo overlaying a lumen and thereby forming a flat disc, we instead have the embryo directly overlaying the yolk and then giving rise to this hollow structure, which raises the question, what about other reptiles? So is the anole the odd one out, and do all other reptiles develop like chicken? Or do all other reptiles develop like the anoles, and is the chicken the representative for the birds? Which brings me to the conclusion of the anoles and towards the next species, the veiled chameleon, which you see, or one of them you see here. And this is a collaboration with Paul Trainer and Natasha Schilo at Stowers Institute. Yeah. So the veiled chameleon is a bit different. It roughly has 40 days from copulation to oviposition. There are 50 to 90 embryos per clutch, which makes it very amenable for quantitative measurements. They're all roughly time matched. And interestingly, and very rarely for squamates, upon oviposition, veiled chameleon embryos are in the early stages of gastrulation. Most squamates, when they lay their eggs, their embryos are at different stages of organogenesis or late neurulation. The pre-gastrulation development, so the pre-oviposition development, is for the most part unknown. The last two studies were performed by Carl Peter in 1934 and 1935. You see one of them, which is both very beautiful studies, but they might need some updating. When I reached out and established this collaboration initially, Natasha had offered me to dissect a few 0 DPOs, so embryos collected at the day of oviposition, and do some histology for me. And then a while later, she emailed me and said, "We should have a chat. She has the first embryos ready." And she showed them to me and said, "Okay, Antonia, I don't know what I'm looking at, but this is really not a chicken." And I almost got a heart attack because what I was looking at was a human Carnegie stage 5C embryo. Do you know which one is the human and which one is the chameleon? Well, this is the human, this is the chameleon. Both of them share a multinucleated trophoblast, which you can see here and here. They both share an amnion, here, and already quite thinned out here. The epiblast lumen, which again reptiles are not supposed to have, because they're supposed to be flat. The pseudostratified epiblast and the hypoblast. So, how do we get to that morphology? I thought it's going to be fairly straightforward. Famous last words. Because we knew that chameleons are supposed to lay their eggs 40 days post-fertilization. So I thought, I'm just going to take one female at 20 days and then we see where we're at and then we work our way in either direction. Yeah, we recovered this at 20 days post-copulation, which is the ovary, and down here you see the uterine, or one of the uterine horns. We collected this at 20 days. We collected the same structure at 30 days, at 32 days, and at 37 days. At the same time, one chameleon laid their eggs after 19 days. So we were a bit confused. What we did notice, though, was this shadow here, which, if you do histology of that, reveals an empty sphere under a single cell layer. If you do TEM on that, which we did, and Melania, who did this for us, told me at some point that this was one of the most boring structures she had ever sectioned because it was completely empty, except for one tiny bit in the middle, which you can see here, which looks like aligning chromosomes. The outside, if you zoom in onto these structures here, you can find nuclear pore complexes, the Golgi, mitochondria, and here you have a double membrane with small holes. So this seems to be the germinal vesicle, so the nucleus of roughly 300 micron of the chameleon egg. And we do know that chameleons have 12 or 24 chromosomes. These ones are very clearly more than 12, so that we might have a diploid set here. And indeed, if we now go for DAPI staining, so the DNA, we can see when we go through that sphere that we have a DAPI light up in the middle of it. Which leads to the question whether this is a diploid set, is that now already fertilized, and do they do some sort of diapause? Or is that not fertilized yet? And for this, we took advantage of the fact that these spheres are massive. They are 300 micron. You can see them with bare eyes if you want to, and you can dissect them. So I dissected every single nucleus of every clutch that we got, and extracted the DNA. And here you can see two control PCRs. Well, here first then you see male, then female empty control, and then the germinal vesicles, and then just ovarian tissue, and that for 3 different time points. And you can see we got DNA for all of them. If we now run this for 2 sex-determining marker genes, 2 male sex-determining marker genes, we can see only the male control lighting up. If these embryos were already fertilized, half of them should be male, and thereby, if we pool all of them, we should see male DNA. But this being completely clean, showing that all of them are female, so they must be not fertilized yet. So, overall conclusion, they store sperm, which we did not expect. Even though many other reptiles do it, they were supposed not to do it. So, how, with a finite number of chameleons, and not knowing whether they are starting embryo development or not, do we find out whether we do sacrifice a female to then look at the right stage? The answer is ultrasound. What we did notice is that at the time point where embryo development starts, the eggshell starts to be deposited. And the eggshell has a higher density than the inner organs, which is visible under ultrasound. So here you can see the ultrasound protocol. This is Rick, who's holding one of the chameleons, and they might hiss a bit, but actually don't mind the procedure much. You add some hydrogel and then do the ultrasound. And we started doing that every 48 hours for the entire female colony, and were able to thereby track follicle and then egg development. Up here you see the ovaries with very small follicles. They're all the same magnification now, what I'm showing, and now much larger. You can see again the single follicles. And I would like you to focus on this shadow, on the space between these 2 follicles. The next, so you can already see it here. There is you have a light line, a dark line, a gray shadow, and again a lighter line. That gray shadow is the initiation of the eggshell, which you can see now much stronger here, here, and here, and then eventually very prominent. So this allowed us to trace development and thereby decide when we want to collect the embryos, and now being ready and set up to answer this question, which is, how do we get to this shape? We first managed to describe the cleavage divisions, which are quite similar to what we found both in the anoles and also what was been described in chicken. You can see here, the first cleavage is, again, not fully cellularized yet. And then you can see here very nicely the border between the embryonic and the upembryonic part of the embryonic disc at the stage. If you do IF, fluorescent stains on that structure, you can see again that plate. And if you then look for DAPI, so DNA, and zoom in at a deeper Z, you can see these tiny dots. And here zoom in. These are the nuclei of the emerging blastomeres. You can see actually here you have aligned chromosomes ready to be divide. So we have the cleavages that are very similar to other squamates. We also then have very small nuclei at the inside and getting larger towards the outside, which we can trace very nicely. You can see here that the further you go outside, you first have them interspaced, small cells with larger cells, and eventually the larger cells get even larger until towards the end you still have full cleavage furrows and not full cellularization happening. So this is just upon the completion of cleavage divisions. So now the question would be, do we have something similar to what happens in humans? So do we now have a mass of cells form that then hollows, maybe through fluid pumping, maybe through cell death? We don't. We actually have this structure. And you can see here, so the dorsal side, this is the outer surface, this is the inner surface, so this is facing the yolk. You see two layers, but these are made of several cells, several cell layers. The dorsal side is the epiblast. The ventral side is the hypoblast. And you can see, when you focus on it, you can see these tiny shoulders. In the next stage, these shoulders started to move in. So they start constricting and they were forcing themselves over that elevated epiblast. Until eventually they're almost close. You can see here it's almost closed until until they then form a radially symmetric structure. And if you look at this now from that perspective, so we're focusing from here, you can see that we have something that looks a little bit like a purse string. Maybe some people of you find this reminiscent of wound healing or neurulation, where you have a supracellular actin cable that is pulling together the neural folds. At the same time, if you look at laminin, the basal membrane has not fully formed yet, which I will explain later why this is important. So once the lumen is completely formed, you can walk here from one part of the embryo through. You have the lumen opening and then closing again. The embryo is radially symmetric. And at this stage, now we're focusing on it from the ventral side, you can see that we have a round structure and we have the basal membrane fully formed. This would be the same if you wanted to look at a mouse embryo. Following implantation, you have the egg cylinder, the basal membrane is fully formed, and then the embryo undergoes anterior-posterior patterning. And similarly here, just a bit later, you now have the breakage of the basal membrane, you can see here, which is then the site of gastulation. Gastulation marker Brachyury in green is overlaying the site of the broken basal membrane here in white. And if you look at this from a different angle, you can see here the double nucleated trophoblast, the amnion, the epiblast, and you see this structure here. This is the site of gastulation. So now we understood how the embryo forms, or gives rise to the structure. And even though this is different from how the human embryo is doing it, we might still have now the similar signaling pathways that are now used to make the embryo undergo anterior-posterior patterning. So we were looking first for anterior markers, and here's Cerberus, which is one of the master regulators of anteriorization in supposedly everywhere. And you can see it first everywhere. which is quite similar to the human embryo actually. The human embryo is expressing Cerberus in the entirety of the hypoblast, and then it segregates it towards one side. So we were quite excited when we saw this, because we thought maybe now the Cerberus is going to be segregated towards the anterior. First of all, it became stronger, which is also fine. But then we saw this. And it was very puzzling, because this part here, this little nose, this is the posterior, this is the site of gustulation. We have the anterior marker Cerberus overlaying the site of gustulation, which is the only part of the embryo where it shouldn't be. So we thought maybe it's just not Cerberus, maybe we need to look at lefty, which is the other of the big two anterior markers. And lefty was first not expressed at all. Lefty is also coming on a bit later in mouse embryos and also in humans. It's not quite as strong as Cerberus, so not that surprising. But then it didn't come on at a later stage, and it wasn't there at gustulation at all. So we thought maybe the gene is not active. So we stained left-right patterning stages, and we saw that lefty is active. It is just not expressed at these stages, which means that anterior-posterior patterning in the chameleon is going via a different route than what is supposed to be the canonical pathways for anterior-posterior patterning, which you can see here. You would here have the distal visceral endoderm that is expressing Cerberus and lefty at the tip, and then they migrate towards one side, Cerberus lefty. That's what the textbooks say. Thus far for anterior-posterior patterning. Another marker, which, if you remember, would come on here, which would be nodal. Nodal would be in the posterior epiblast, and then signal down to tell these cells to become anterior, and then eventually to overlay the site of gustulation. And nodal was initially not expressed, but then it becomes expressed only in the middle of the embryo, at the tip, which was quite exciting because this shows that the embryo at this stage is radially symmetric. And then it's segregated towards the posterior. So we thought that this was actually quite nice. And then we hoped that if we now stain for Burkeri, which is the marker of gustulation, because it's a mesoderm marker, that Burkeri might follow the same pattern. However, it did not. Burkeri was first not expressed, but then we saw this. This is at a stage where the lumen is just forming, so the embryo has these elevated shoulders that are constricting. And once they are completely constricted and close the lumen, we have the entire embryo positive for Burkeri. And then it segregates to be on two sides, until eventually it is at the posterior. Burkeri is not supposed to do that. It is not supposed to be active at any time before gustulation. It is never supposed to be a ring, in amniotes at least. So, which you can see here, just as a reminder, this is a mouse. You can see Burkeri being expressed at the border between epiblast and profectoderm, and then eventually being being expressed in the primitive streak, but never you can see a ring. So it was quite puzzling, which leads me to my conclusions on the chameleon part. It's just that clearly epiblast lumen formation is not a mammal-specific trait. Whale chameleons form an epiblast lumen via a novel mechanism that includes a purse string, supracellular actin cable, and circular Burkeri expression prior to anterior-posterior patterning. We also report that anterior-posterior patterning exhibits divergences from the canonical patterning in amniotes. So, what do these findings mean for other non-avian reptiles? I've already shown you the anoles that are quite different from what I've shown you in the chameleons. So clearly, non-avian reptiles are not we cannot just take one shape to fit them all. We need to look at all of the different families. And I want to give you a brief look through old literature. And here you can see an old paper, an old French paper from 1968 from Weber and Dufouré, where you can see the cleavage divisions, then full cellularization, and then this is the initiation of gustulation. And you can see that it has never been reported as being hollow or anything else. And also, you might, this is just one photograph. They did not do any sections or something like that. But you can see some shadows which suggest that this might actually also be a bit elevated. And not being a flat disc either, and also actually being here teardrop shaped, which is different from what we shown in the anoles, and also very different from what we found in the chameleons. If you look at a different species, the common lizard, Zootrocha vivipara, which was staged by Dufouré and Weber in 1961, we can see this stage here, initiation of gustulation, which also doesn't look as flat as a chicken. It also doesn't look like either of the species I've shown you before. But instead actually seems to be a bit tunnel-like, where you have the anterior-posterior axis straight, but generally elevated. So this speaks for quite a bit of diversity in reptile embryogenesis, which as of now was underappreciated. Which brings me to the last point, which is the epiblast lumen, that none of these species has exhibited, but Mabuya does. This is a paper focusing on the placental structures of Mabuya, but here you can see the embryo. And I think it's very visible for everyone that this epiblast is surrounding a central cavity, which is a lumen. They also draw it into here, even if they don't focus much on it, showing that also luminogenesis does not seem to be, A, doesn't seem to be mammal-specific, but B, also does not seem to be involved only once. Mabuya and chameleons are quite distant from each other. So maybe similar to live birth, which has evolved multiple times in reptiles, luminogenesis might also have evolved multiple times in reptiles, which then of course opens the question of how do they form their lumen. Do they form it similarly to human embryos because of that entire blastocyst-like structure, or via a completely novel mechanism? With this, I would like to come almost to the end of my talk, which is why I'm here. I'm working with Litash to stage some pre-oviposition Pachyndactylus tigrinus, because geckos are a sister family to all squamates. You can see them up here, and I find that quite fascinating because it would be interesting to see, well, what morphology they have adopted. And with this, I would like to finish with telling you what else I'm doing. So generally, we have covered quite a bit of reptiles in this talk, and there are a few more coming. So I'm going to get some turtles soon in North Carolina. But this doesn't enable me to cover, to understand how amniotes generally how embryogenesis works and generally, and how diverse it is. For this, I need other species as well. So we have here echidnas from Ollie Griffith at Macquarie University in Sydney, who also give me access to donuts and wallabies, which would be marsupials. And then I have a collaboration at the German Academy of Health and Environment to access deer, roe deer, fallow deer, and boars and foxes and hares. And I also established a collaboration with Colombia to access hippopotami. And yeah, generally, I'm always interested in more collaborations, and I can never say no to a new species, so let me know if you have something fun. And with this, I would like to thank all my collaborators, my mentor, Susanna, in Oxford, my postdoc supervisor Florian, who was incredibly supportive of me starting my independent project, Tony Gamble for the gift of 140 morning geckos, Lukasz, Bara and Tomasz here, Paul and Natasha at the trainer lab, and Cornare, which is my collaboration to access hippos. Joachim Redemann for his tireless efforts to give me access to the deer, roe deer, fallow deer in Germany. Tom for the anoles and soon to be corn snakes. Bonnie and Ollie, and my funding body, All Souls College and the John Fell Fund in Oxford, and the funding bodies of all of my collaborators. And with this, I would be happy to take any questions.